So today we have a very special researcher with us, Dr. Rupkatha, who will be joining from the Scripps Research Institute, Florida. She is working as a postdoctoral fellow in the Ron Davis Lab in Scripps Research, Florida, where she is working to develop mitochondria-based therapeutic drugs primarily for Alzheimer's disease.
Neurodegenerative diseases are a major threat to human health, especially because it affects millions of people worldwide. The most common forms of neurodegenerative diseases include Alzheimer's disease, Parkinson's disease, and Huntington's disease. So 95% of the other cases of AD are the sporadic AD cases. This is also known as late onset Alzheimer's disease. If you look at the brain of an Alzheimer's disease infected individual, you'll see that the area covered by the brain is much smaller. The brain looks tiny as whole. Organelles, RNA, they are all packaged together in compartments, which are called vesicles. These are like globular shaped structure. And these motor proteins get attached to the vesicles and they walk along the microtubule tracks. So we can say that axons are basically like highways inside the cell. Highly bright structure, this is the brain. And these lines that you see that are coming out of the brain, these are the nerves. We are seeing them as dotted, right? So these dots are basically because the Huntington protein has been tagged with red fluorescent protein. That's why it helps us to visualize. Previously, scientists had thought that 𝗥𝘂𝗽𝗵𝗫 perhaps phosphorylates motor proteins, like the motors that I talked about, kinesin and dynein. However, there is no proof if actually GSK phosphorylates. And I just cleaved that region of presenilin, transport was all messed up. And yeah, we saw those traffic jams in the nerves. So to know more, I generated a kinesin mutant fly, which is phosphodeficient, which means that this kinesin cannot be modified by 𝗥𝘂𝗽𝗵𝗫. So they screened a battery of compounds, including alvarine, orphanedrine, yohimbine. So these were the very potential targets, alvarine, orphanedrine, and yohimbine. And these were selected to be tested at the next level. So the next level is where I come in. So I test these drugs in Alzheimer's disease mice models. And we expect that in AD mice model, the mitochondria would be highly defective. And we also expect that these drugs that we shortlisted, these drugs will reverse the mitochondrial health back to normal, at least even if not completely, at least maybe it would be a little bit efficient. And then the scientist moves the mice and sees how fast it goes back to that platform that it was conditioned to. And now I'm in Florida, completely, entirely different climate, watching alligators. Here you see this video my friend took 2 weeks back, this alligator.
Is how we modify the gene, like
in Drosophila?
Yeah.
Okay. So what happens is, so there's this system called UAS-GAL4. Are you aware of that system? Else I'll go in deep details.
No, I'm not aware, sorry.
Okay. So what happens is you have two flies, right? So one fly expresses this driver called GAL4. So this is an yeast driver. You can look it up. You can Google it and learn more about it. And so we have one fly expressing GAL4, and we have another fly expressing UAS, and the gene of interest is just downstream of UAS. So what happens in yeast is that GAL4 binds to this upstream activating sequence, or UAS, and causes overexpression of the gene that is downstream of the upstream activating sequence, right? So what happens is you have one fly with GAL4, one fly with UAS, and your gene of interest. You cross these flies, you mate these flies, and the offsprings will have both GAL4 and UAS. So GAL4 will bind to UAS and will cause overexpression of the gene of interest. So if you can cause overexpression, and then you can, in the gene of interest region, you can add RNAi so that there is expression of RNA, like say RNAi against some protein. and like downregulation of that protein, basically, knocking down the protein and so on. And also, I should mention that this GAL4 and UAS, these are lines which are all you introduce into the flies. The flies don't have GAL4. It's like based on GAL4, yeast. And also, it's very easy because if you have GAL4 under a tissue specific promoter, for example, if you have GAL4 under a brain specific promoter, you can target GAL4 expression only in brain. So for example, in my lab, we used to use APPL promoter, APP-like promoter, which is expressed in the central nervous system. So what happens is, if you have APPL promoter before GAL4, GAL4 will be expressed only in the brain. And in the offspring, GAL4 will bind to UAS only in the brain and cause overexpression of this downstream gene only in the brain. So that is how we modify. So research, as I said in my video, it's all about relentless forward progress. It's hard. I mean, it's nice like to hear that, okay, she's a scientist, she's working on this, she's working on that, but it requires a lot of motivation, and you have to fix your mind that, okay, this is what I want, and everything should come from within. Sometimes there will be days where you won't feel like doing an experiment. Your experiments won't work. You will get negative results, but don't lose hope. Maybe the hypothesis is wrong. Troubleshoot the hypothesis. Maybe you did something wrong. Go back, check what you did wrong, and I'm sure you'll find an answer. I had a lot of problems in my lab. I saw people with problems, but yeah, research, as fancy as it sounds, it comes with a lot of thinking. You have to think. You can't just do blindly experiments blindly. You have to think about it. Okay, is this rational? Is this logical, the thing that I'm doing? And then move forward. And then in the end, it is rewarding. That I can promise. It's tough, but the end results are always good. Yeah. So if you are up for it, you should go do it, because we need more researchers. And with COVID and all, we know that, right? We need more researchers. Yes.
Yeah. You're a researcher on web. We try to bring real researchers doing research that brings forward these advances in technology, medicine, and so on, and showcase them to the world so that every research enthusiast out there gets to know where the world is going forward and what these amazing researchers are doing.